I'm from Stanford also, and I'm from the Department of Genetics. So what I want to tell you a little bit about is how we see personalized medicine at Stanford, and perhaps this is going to be a similar story across other academic centers like ourselves. So one thing that differentiates personalized medicine for us is we look at this as a collection of data coming from different sources. So one is the research sources right now, which is a collection of omics, microbiome data, and more recently, data that is being collected from sensors and apps. Then we also have, because we are part of our system is our hospital system, we also get clinical data. It's your family history, your diagnosis, different types of imaging data, your laboratory tests, your prescriptions. Part of this data moves very quickly, so your sensors are collecting at a 1-second interval, your microbiome changes at a few hours interval, and hopefully your omics is changing much slower than that. Microbiome is actually extremely interesting because there is a lot of data that you can collect over a period of time. So let's say you're sampling a human being every month, you can potentially easily collect 5 to 10 terabytes of data just on one person collecting on a per month basis. So ballpark calculation, 2 petabytes of data for a human being, and lots if you include the entire population. So I'm going to give you a very high-level overview of what are the different types of projects that happen, and I'm going to start from the clinical service because that's what we usually consider closest to personalized medicine. So we have a new program that has come up in our hospital system, including our adult and the children's hospital, that's looking at whole genome data, and an earlier speaker mentioned that we have 4 billion bases. So it's looking at all 4 billion bases to actually figure out what's wrong with you. This is expected to impact lives of about 5,000 patients per year. 5,000 patients per year translates to about 5 petabytes of data per year. So not a whole lot, but I think this is a good start. There are several programs like this at Stanford, but perhaps this is at the largest scale. Then there are several programs, much like the MVP that previous speaker talked about, is biomarker discovery, where we actually look at a group of patients, and also that typically includes controls, and you try and find out what's wrong with your group of patients that you don't see in your controls. We will be getting some data from our collaboration with VA, which will this year include 2,000 whole genomes and 20,000 exomes. This is a very exciting project for us. And going between a biomarker discovery and clinical service, there is a bit of gap, but this is typically what you would do in a pharmaceutical industry, where you will find something, you'll go back, develop mouse models, and then you'll figure out what the drugs are that you can give to the patient. Between biomarker discovery and basic science, so there are, again, if you're doing 10 biomarker discovery projects, you're probably doing hundreds of basic science research at Stanford. One of the largest projects is ENCODE that we have a poster on outside. Stanford is one of the largest production labs. It is also a data coordination center. Again, going from basic research to biomarker discovery is understanding what the function of your genome is. That eventually translates into which genes are disease-causing potentially. So we look at this entire process end-to-end and think of this as personalized medicine, and that's the kind of work that's happening at Stanford. So from a technology point of view, we have a lot of challenges, and part of the challenges is we have a whole lot of data, and there are lots of privacy concerns around this data. And as some of the speakers you have heard before today, data in our case is extremely, extremely important. We can change the compute stack, but the data is something that we want to hold on to, and we want to come back to this data again and again. So some of the challenges we are seeing now, and we are addressing this, one is being able to process these hundreds of thousands of genomes and do it systematically in a manner that we can potentially do in a traceable manner, because these things have clinical implications. So I would posit that the ecosystem in being able to do this kind of data analysis is fairly immature right now, And the reason for that is we are actually trying to do this analysis at different systems. We don't have one large monolithic system where we are collecting data. Our data is scattered across multiple systems, and we want to do this kind of traceable computing. Secure collaboration is an extremely difficult problem because the data is in silos. We haven't solved this problem yet. We can only solve this in the context of Stanford. Real-time data mining, we briefly discussed BigQuery, but this is, again, an immature system. There is lots of work to be done. And finally, being able to combine knowledge bases from hundreds of different sources. This is an amazingly difficult problem. We call it like the busy bee. You sit around and you keep doing ETLs over time. So thank you very much.